So I am the co-founder of a nonprofit, which is based in Ithaca, New York. So my co-founder is Alison Wilson, and we're both biologists, molecular biologists, geneticists, and we mostly study plants. And what we're interested in doing, let's see if I can move the next slide. So what most people know us for is a website called Independent Science News. And what our nonprofit does is we take kind of deep dives into areas that look really interesting to us. And sometimes they end up as news items or kind of in-depth write-ups on this website. And they're all about food and agriculture. And sometimes they end up in the peer-reviewed literature. So, we try to publish scientific papers when a lot of referencing and a lot of kind of deep scientific kind of bolstering is required for what we want to show. Then we put those in the peer-reviewed literature. And that's what I'm going to talk about tonight, is some of that work, but try to make it pretty understandable. The second kind of framing for our work is the centrality of food and agriculture. As Alex has alluded to a little bit, we see food and agriculture as the nexus of all kinds of social and ecological problems. So we see it as one of the primary drivers of biodiversity loss, of problems with human health, for example. One of the primary determinants of the quality of the water that we have, or of coral reefs. I made a little list here of the things that are extremely negatively affected by industrialized agriculture, but at the same time can be remedied by local, organic, regenerative, agroecological types of agriculture. And what's really interesting about our current political situation is a lot of other people are seeing these connections too, and they're becoming members of the food movement, right? So what these people are seeing is that we can remedy all these, what are considered by people who are not members of the food movement as intractable and inseparable and phenomenally complicated and diverse problems. They can be solved through food and agriculture. What I say to people is, if you want to solve our climate issues, if you want to solve people's health, you have to look primarily to the food system to help you do that. And if you do look to the food system, you can solve all these problems simultaneously. They all simultaneously can be solved by the same solutions. But if you decide to ignore the issue of food and agriculture, you will make basically no progress solving any of them. So this is the kind of logic that we use in our project and have kind of developed over the last few years to guide our behaviors. And what I'm going to do tonight is talk a little bit about an aspect of GMOs that came to our attention actually as a result of somebody who lives near Melbourne. I don't actually know because I've never met her in person. But she's a woman from Melbourne who kept sending me packets of information about the toxins that GMO manufacturers put into crops. So these are toxins that are intended to kill insects that they put into the crop. And that, as I learned at the conference I was at last week, the academics and the companies would like to change their names away from something called toxin because they're concerned that people think that if they keep calling them toxins, they will draw certain conclusions. So this is the title of the paper that I gave at the scientific conference last week. So, my initial reason for coming to Australia is that held last week, 10 days ago, was the Society for Invertebrate Pathology, so this is killing insects, basically, was having this conference in which this is the foremost place in which you discuss the functions of these proteins. So and this is a picture. What I show you, what this diagram shows is the nature of one of the natural versions of these proteins. So what I wanted to talk about was the difference between the naturally occurring versions of these toxins and the ones produced within GMO crops. Because the story of the companies is that these are toxins that are used by organic farmers, which is true. They're used in forestry, they're used in conventional agriculture, and for the most part, they've not been associated with human health consequences. And so the conclusion of the regulators has been, if you put these same proteins into a plant, then you will not find any worse health consequences. And so what we wanted to do is, helped by Madeline Love, this lady from near Melbourne, is, she drove us to think very carefully about the nature of the differences that companies had incorporated when they put these proteins into plants. And I'll tell you about that in due course. So just to go back a little bit, these toxins are envisaged by many people as replacements for synthetic insecticides that also have numerous health consequences. We either know or suspect. So the story that is fed, especially to the US public, is that there's been a huge decline in pesticide use because of the toxins produced by the GMO crops, the insecticidal crops produced by Monsanto and by Dow. So we're talking mostly corn, but we're also including some soybeans. We're also talking about inside, there's a new crop which they've introduced to Bangladesh called eggplant, which also now has these cryotoxins in. We're also talking about cotton. In Australia, cotton is grown, most cotton is grown, and it contains these crops. But that food, most people don't know, can also end up in the human food chain. So certain parts of sausages, for example, are made with lint from cotton, for example. Cottonseed oil is used quite commonly in the human food chain. So the big story of the companies is that they're saving the planet by reducing pesticide use, and they roll out these figures. And the problem with these figures is that USDA decided not to count it's true. So it's true. It's a fact that there's a less use of sprays on these crops. What the USDA, US Department of Agriculture, who keeps the data on pesticide use in the US, doesn't tell the journalists who come and ask them how much pesticide use there is. They don't tell them one thing, which is that neonicotinoids are now used as seed treatments, right? So these are preventative pesticides. So you put them on the seed, and then the seed is protected because inside it, systemically spreading inside that crop, is an insecticide that kills the insects that would feed on it. So, but USDA is not counting that data, right? They interpret sprays very narrowly, right? So they don't count the insecticides that are being used, that have essentially replaced many of these sprays. So the other thing they don't count is the cryotoxins, the Bt toxins that are put into these crops, right? So imagine this toxin is produced all the time inside the crop. So it is there 24 hours a day. It's produced in the leaves and the roots and the pollen and the seeds that you eat, for example, and corn. So if you buy GMO corn, it will contain maize. This is maize. Corn is maize, in case anybody's confused. So these insecticides are in all these crops. What you also should know is that these insecticides that are in these crops, it used to be the first generation had one transgene producing this insecticide. The latest versions of the maize, corn that's used in the US has 6 different transgenes in it. So you see an escalation of the use of these toxins. And each one is a little bit different, as we'll see. And so what's happening is an escalation of insecticide use in American agriculture, but also in agriculture of other places where these toxins are used. And this is just the title of the paper. And in the middle, you can see the name of, this is Madeline who started this project. And this is a collaborator we had, the battery's going. Angelica Hillbeck is a collaborator that we had, and she lives in Switzerland. So, and this is just the name of the paper, just so you can see that we wrote a scientific paper. And the data set that we used here is actually Fazan's approvals, because Madeline is from Australia. She wanted to basically understand what was being approved in Australia, partly because this is relevant to Australians, but also because it's kind of reflective of what is going on in world agriculture. Like what you are importing into this country is essentially this sort of generic sampling of what's used in global agriculture, at least in the world of commodity crops. So, the story offered by the regulators, and it is offered by the companies such as Dow and Monsanto, who make these crops, is that these toxins are natural. You can see in these, I can no longer point at the screen, but what essentially, you can see they're using the word natural, or it's used in agriculture and so on and so forth. So they have a narrative that what they're producing is exactly the same as what's been used, basically in very small quantities by organic farmers and so on and so forth. So in GMO agriculture, produced inside the crop, we're talking about massive quantities of these toxins. What organic farmers are using them, but basically in small quantities. So let me just explain a little bit about these toxins. They're either called cryotoxins or Bt toxins. It's essentially the same thing. Bt stands for Bacillus thuringiensis, and that is the name of the bacterium from which they come. And what you will see, if you look at this slide for long enough, is that essentially those are in the middle. They're also called delta endotoxins, just add to the complication, but we're also talking about the same thing. And they're crystals. They're formed by the bacterium as a crystal. And these crystals basically enter the insect, and the bacterium basically splits open. The spores of the bacterium split open, and the crystal is dissolved by the chemical and enzymatic conditions inside the bacterium. And the crystal, the toxin that's kind of broken, basically you get this kind of steady breakdown of the toxin, and the toxin eventually makes holes in the membranes of the insect's gut. And then that insect's gut basically explodes. So it's kind of like every cell, imagine every cell you prick, like it's like a balloon, and you prick each cell. As soon as you equilibrate the air on the inside and the outside, or in this case the water on the inside and the outside, the cell explodes because the toxin has made a hole. What's interesting about these toxins is that all biological organisms, except for viruses, are full of membranes. Right? We have membranes in our gut. You probably know. You have membranes surround every single cell that you possess. And the same is true for the insects. So you have a family of toxins that can harm butterflies, that can harm Diptera mosquitoes, by the way, that can harm coleopterans. Rhabditidae here are nematodes. You also have snails, for example, or ants, or aphids that can be harmed by these toxins. And you also have demonstrated in the scientific literature the reality that these toxins can also harm human cells. Right? Can also make holes in human cells. And what's interesting about the regulatory situation here is that the companies are saying, we're taking a toxin, that is, for example, this one, Cry1A1. So each one, there's a whole list of toxins here, each of which is considered to be specific to one of these insect groups. So we have a complicated situation in which very specific toxins kill specific families of insects, but at the same time the whole family is responsible for killing a huge number of different organisms. But each toxin is considered by the regulators and by the companies to be very specific to that group. And this specificity is the basis of approvals. Because if the whole problem of sprayed-on insecticides is they kill everything, pretty much. Right? There's hardly any species that are unaffected by these insecticides. What the argument that the companies are making is that these insecticides that they put inside their crops are very specific. They will not harm you because they're specific to mosquitoes, or they're specific to lepidopterans, for example. So they're only killing the pests. So this is one of the, essentially the basic argument of the biotech industry, agribusiness, that they have this understanding, this deep understanding that these toxins will harm only the organisms that they are intended for, and will not harm anything else. So the fact they get into the water supply, the fact they're in the pollen and blowing all over the countryside, the fact that they're in the soil and so on and so forth is of no consequence, right? This is the argument of the industry, the agribusiness and farmers and so on and so forth. The problem is that you have this family of toxins that in the round can kill all these organisms. And the basic reason is because they have this mode of toxicity, which is to make holes in membranes. So, the companies arguing to the regulators in Australia and other places that these are natural toxins. Because they have a history of safe use, they're fine inside their crops. Accepting that the companies have made changes to the molecules. So they have shortened the molecules, for example. So you might think, oh, shorter is a good thing, right? Well, shorter is not a good thing. Because if you imagine that these molecules are the biological equivalent of a nuclear weapon, right? They can basically kill any organism in principle, because all they do is make holes in membranes. Any organism that has membranes can be harmed by these toxins. So think about the bacterium that holds onto these toxins in the first place, the naturally occurring bacterium. Why is it holding these toxins into a crystal? It's putting them into a crystal because it doesn't want that toxin to escape and destroy its own membrane, right? So it has set up the situation so that this equivalent of a nuclear weapon cannot go off inside itself. You wouldn't want that, right? So these organisms, the parallel with nuclear weapons is really powerful, because you think about countries that own nuclear weapons. The last thing they want is for that nuclear weapon to go off on their own territory. So you build in all these safety features which make it bigger, and which make, for example, there's a passcode, and there's a metal box on it, and there's a timer, and there's a height device, and there's all kinds of things that you add onto it as kind of safety features, right? And what's happening when the companies make these chemicals is they're removing the safety features, okay? So a truncation means that toxin is more dangerous because it's smaller. It's not less dangerous because it's smaller. So the companies also have made other changes. So they make additions to the protein, which they don't explain the reasons for these changes, for the most part, to the regulators. So they add, the CTP is a protein sequence that is, essentially it's a targeting sequence. It stands for chloroplast transit peptide. So if people know, we're talking about, this is in plants. The plants have chloroplast. Chloroplast is the photosynthetic kind of organ of the cell. So this is to move these toxins into that organ. We don't know why they do that, right? But we can make a guess that I'll hazard to you in just a second. So they also add random sequences. This is interesting. A company making a highly specific and precise GMO has told the regulator that inserted into that sequence into that transgene is a random sequence. Right? This is, as a regulator, if I was allowed to be a regulator, I would never allow a company to put a random sequence into a toxin, right? This is just, it's molecular madness, right? But they did it. But it kind of gives you a little insight, right? Maybe we'll go there tonight, into the mindset of the regulators that exist, not just in Australia, but also abroad. Companies also make amino acid substitutions. So amino acids are the components of proteins. So they make changes to the toxin. But they don't tell the regulator the purpose of these changes. They just say, we've made them. It's still almost a natural protein, almost indistinguishable. The companies also, if you think about a protein made inside a plant, Normally a protein made inside a plant is a soluble molecule. The natural form is a crystal. And what I've told you about the crystal is basically a way of preventing the toxin from being activated. If the plant is producing a soluble version, that means it's more likely to be active inside the organism. So you have all these changes which are a consequence of these toxins being produced inside the plant. So we can ask ourselves, what is the reason why the companies make all these changes? Right? We can hazard a reasonable guess that it's to make their product more effective. Right? It's to kill more bugs and have an effect on a wider range of species. In other words, they're reducing the specificity. And what I wrote at the bottom is that this is the theoretical expectation of what these companies are doing. No, companies don't normally make their product less effective. So we wanted to know more about this, and Madeline from Melbourne found a patent, Monsanto patent, on the same toxins that Monsanto has been putting into their corn plants, MON 863. This patent is describing the exact same molecules that they told the regulator as identical, or nearly, as near as makes no difference, identical to the natural toxin. In the patent, they're calling these toxins supertoxins. And they're calling them supertoxins, and the quote is at the bottom, they have the combined advantages of increased insecticidal activity, right, which means basically the toxin works better, and they explain what changes they made, and you can expect that would make the toxin work better, and also that the toxin is active against a broader range of organisms. Now, they don't test that broader range of organisms that very widely, but think about what a broader range of organisms is. That includes potentially mammals, people, cattle, and so on. But they don't go there because they're not doing the test, right? But they do know that this toxin is more broadly active. So I'm going to stop talking about those toxins. Are we nearly up for half an hour? Can I talk for a couple of minutes?
Another 10 minutes.
Yeah, okay, great. So, I've given you this doom and gloom story, right? It's not very happy thing about toxins that there's a strong possibility, right? Making holes in your gut and the guts of other organisms too. Now, we don't have too much evidence about what happens inside your specific gut. But I was attending, when I was in Rockhampton last week, I gave a presentation alongside an Australian researcher called Judy Carman, who is definitely worth knowing about. And she has done a series of studies, basically independent of the kind of official research system, at least in part. She's a PhD, she's an epidemiologist, she's a public health scientist, and so forth. But she has managed to organize the funds to study some of these GMOs and do experiments in which you have a control set. And she did experiments on pigs and experiments on rats, totally separately from each other, and feeding slightly different GMO corns. But what she observed in her experiments was inflammation of the intestines. She observed the sort of separation between cells that you shouldn't observe when you have a gut, right? Your gut should be kind of tight, and the cells are kind of coming apart at the edges. And this was a consequence of the feeding of these GMO crops. And she can't necessarily say that these cryotoxins are responsible for that because she doesn't quite have the right controls and so on and so forth. But essentially, she has controls which are not, where the animals, they're not eating any of these toxins. And those animals look pretty healthy. They're being fed, kind of conventional food from commodity agriculture. But when she feeds them these corns, basically corn, containing these toxins, she sees this inflammation. You can see that the guts are just red. Unfortunately, they dissect the animals, but you see these guts being much redder, for example. And farmers are reporting health problems among their animals, she says. And I don't know, because I don't talk to too many farmers and conventional farmers and so forth. But this is her account of the issues. So, we can build up a scary picture. When we put it together with Alex's concerns, and mine too, right? I don't have gut problems, but I know people who do. So there's a puzzle that could easily be put together here and should be causing alarm signals to be raised by regulators. But I was at this conference last week, and FSANZ is the industry body, or the government body rather, that should protect you from the health consequences of GMOs. We had a week-long conference. It was divided into two parts. The first day was a segment organized by Monsanto and Dow. Monsanto and Dow have basically invited other speakers from other companies, such as Bayer, such as BASF, and several others that you probably don't know. And these are all makers of these toxins. And a couple of academics, and FSANZ came to that workshop. They called it a workshop. But FSANZ is Food Safety Australia, New Zealand. FSANZ, Food Standards, Food Standards, Australia, New Zealand. Oh my God, I got that right. Excuse me. I don't want to pass on any misinformation here.
Yeah, no, it's not about safety at all.
Well, yeah, I think they were probably, well, we won't go there. So, the FSANZ came to the Monsanto workshop. And I asked the organizer, I didn't see them on the second day when all the academics are discussing the biological properties of these cryoproteins and the toxins, and, to try to kind of tease out the specificity and all the kind of biological function by which these proteins make their holes in these membranes. And I discovered, and I didn't see them either, that FSANZ wasn't at the rest of that conference, right? So, to me, this is a very alarming thing, right? That your regulator wants to know the view of Monsanto and Dow and BASF and is not interested in the opinions of independent academics, right? They didn't come to my talk. I gave them the same story as this, and I included parts, for example, about how the regulators were the applicants, the companies, were lying to the regulators, right? Basically giving them inaccurate information. And I had people from Dow come up to me afterwards and say, "Oh, we're all nice people, and we would never sell a product that we wouldn't like to eat ourselves or our family to eat." But the problem is that I'm part of the speaking tour. I'm talking about these exact same companies and their concealing of the hazards of dioxins from the public, right? Millions of veterans and hundreds of thousands of babies have been born in Vietnam with birth defects and so on and so forth, because these exact companies concealed information about their own products, right? And those products are Agent Orange. And they don't know that I have access through this poison project to all this astoundingly incriminating information about what they're telling, about what they knew, even going back to the 1940s and 1950s, long before the Agent Orange, Agent Orange was even formulated as a product. So, positive things. There are many, what's great about the food movement is that it's offering solutions to all the problems that we have today. So on the left, we have a book called A Precautionary Tale, which is a really interesting story about a town called Mals in the north of Italy. And Mals is a town that's banned all pesticides, right? It's a farming, rural community. It's 11 villages. It's neither big nor small, I would say. But they have basically banned all pesticides because they saw the advance of industrialized apple agriculture into their communities. So what they decided to do is they decided that this industry was going to destroy their livelihoods, right? They had local breads, they had local grains, they had local small farms, they had a thriving little organic sector, and so on and so forth. There was no way that it could stand up to this, to the advance of industrialized agriculture. So they banned it outright in a citizens' referendum. So this kind of thing is really exciting to me, because these are people drawing the dots and achieving political success. And then this is the story, the picture on the right is the story of SRI rice, right? If you ever worry about whether small scale or local or organic agriculture can feed the world, people who grow SRI rice have a totally different system. It was developed in Madagascar, involves basically little or no application of agricultural chemicals, no fertilizers. They use a little bit of compost, but they've got a system that holds the world record for rice production per acre, 24 tons per hectare rather. So, and this is on a farm that previously was producing 3 tons per hectare, right? So these are people who can really do stuff, and this system of growing rice is spreading around the world. There's now 20 million farmers who use this method of growing rice, because the small farmers love it, right? Because they can devote their rice areas to something else, right? Like spinach or carrots or whatever it is they want to grow. So this is an idea which is really catching on. So I'm going to finish there and maybe just thank the board members and volunteers that we have in our organization, and encourage you to subscribe to our website, right? Our Independent Science News website is a great resource. We don't publish something every day, but you will be routinely astounded by the kind of information that we managed to dig up. I guarantee it. So yes, visit Independent Science News and the Poison Papers and the Bioscience Resource Project if you want to know more about what we're up to on a kind of a more daily basis. So thanks very much. I've gone on plenty long enough, and I will we'll turn over to the next part of our evening. Thank you very much.